The technique of titration is slowly adding an acid to a base or vice versa to neutralize a substance. Volumetric analysis is a method of determining an unknown substance by titrating it against a substance with an accurately known concentration. Using these two methods, the concentration of an unknown solution can be found. To carry out a titration, the standard solution must be prepared. A primary standard solution is the first solution needed in order to carry out the titration, and by which the concentration is accurately known by directly weighing the solid of the substance. In this case, the primary standard is sodium carbonate. The sodium carbonate must be anhydrous. It must be dried at 150 to 180 degrees Celsius in a drying oven for half an hour, and then left to cool in a desiccator containing drying crystals. Measure the weight of the primary standard on an electronic scale for more accuracy, as the electronic scale measures to 3 decimal places. The amount of sodium carbonate can be any amount as long as the mass is known. After writing down the mass, the solid must be dissolved with distilled water. Place the sodium carbonate solid in a clean beaker and using a plastic wash bottle, add about 20 milliliters of distilled water and stir using a plastic stirring rod. Ensure the solid is completely dissolved and the stirring rod is left in the beaker at all times. Then transfer the solution into a clean volumetric flask using a plastic filter funnel. Before transferring the primary standard solution, to ensure that there is no contamination which can affect the end results, the volumetric flask must be rinsed with distilled water several times and discarded in the sink. This is just an extra precaution to make sure the equipment has been washed properly. Otherwise, if you know that the equipment has already been washed thoroughly, then you can skip this step. What is critical that you must do, however, is to rinse the equipment with the solution you are going to put them in several times before actually putting the solution in. This must be done constantly throughout the process to all the equipment where the solution is transferred. This is to ensure that the distilled water doesn't dilute the solution, which will affect the concentration and the end result. However, for this part you do not need to rinse the solution as you will be adding more distilled water later. So after rinsing, pour all of the sodium carbonate solution into the volumetric flask using the plastic filter funnel. To make sure all the solution has transferred, take the distilled water in a wash bottle and rinse around the funnel, the beaker, and the stirring rod, and transfer those into the flask also. To prevent splashing and losing solution, it is a good idea to use a stirring rod to pour the water from the beaker into the funnel. After this is done, make up about 2/3 of the volumetric flask with distilled water. Then put the stopper on the flask and invert continuously to dissolve all the sodium carbonate. Once it has dissolved, fill up the rest of the volumetric flask with distilled water until the bottom of the meniscus is level with the engraved line for an accurate reading. Make sure you get down on eye level with the meniscus to assure that the bottom of it is aligned with the engraved line. Label the flask sodium carbonate and you have made the primary standard. Now you must fill the burette with the HCl solution, but first rinse the burette with distilled water and then the HCl solution several times. Do this by putting a small amount of distilled water or the HCl solution in the burette and putting the opening of the burette over a sink. Hold the burette horizontally so the solution lies across the whole thing and rotate. Then discard in the sink. Also make sure to rinse where the stopcock of the burette is. After rinsing, set up a retort stand with a burette clamp and attach the burette to the burette clamp. Overfill the burette with the HCl solution, going past the highest gradation mark, which is 0.00 milliliters. Wait for all of the solution to run down the sides of the burette, then twist the stopcock to run the excess out in a waste beaker, so that the bottom of the meniscus is aligned on the 0.00 milliliter mark. If there is no white backing behind the gradation marks of the burette, then place a white sheet of paper behind them for an accurate reading. The solution in the burette is called the titrant. To standardize the HCl solution, first you need a pipette. A pipette also requires a special rubber bulb which is attached to the opening of the pipette and can be used to draw up the solution. By using this, you are able to control the flow of the solution as you fill up the pipette. But before you can do anything with the pipette, you need to, yes, that's correct, rinse. So rinse the pipette with distilled water if you need to, and then with the sodium carbonate solution, which is what you are going to use the pipette for, and discard in the sink. On a pipette, there is one special gradation mark, which is the accurate volume of the solution when it is filled up to that mark. Place the pipette with the rubber bulb in the volumetric flask and squeeze the rubber bulb to draw the solution up the pipette. Squeeze gently once the solution is closer to the gradation mark. Ensure that the bottom of the meniscus is aligned with the gradation mark by getting down on eye level for an accurate reading. After this, take an empty conical flask and with distilled water, yup, you guessed it, rinse the conical flask several times and discard in the sink. You only need to wash this with distilled water as you will be adding water during the titration. Once you have done this, take the pipette filled with the solution, place it in the conical flask and squeeze the button that releases the solution until all of the solution is transferred. The conical flask should be held at an angle and the tip of the pipette should be touching the inside wall of the flask so that adhesive forces ensure that all the solution is completely transferred in the flask. The amount of solution in the conical flask is known as the aliquot. Now place the conical flask with the sodium carbonate solution under the burette. Add 2 to 3 drops of a suitable indicator, in this case methyl red, and place a white tile or surface underneath the flask so when the titrant changes color it is easier to see. The color change of the titrant is called the endpoint, and in this case the titrant should change into a light pink color. Now, slowly twist the stopcock and let the titrant run slowly into the conical flask. While this is happening, swirl the flask continuously so the solution is properly mixed in during addition. If any of the solution splashes on the insides of the flask, use the wash bottle to rinse it back into the solution so any excess can mix in. Continue swirling until the titrant changes color and there is no further color change. Accurately read the remaining volume of the titrant on the burette by reading the bottom of the meniscus and record. The volume used up in the titration is called the titer. So the first trial is always a quick, rough trial, which is used to determine the approximate volume needed from the burette. The procedure should be performed 3 more times after this to obtain 3 accurate titers, each within 0.1 milliliters of each other, and making sure to calculate the average of these. Once the endpoint is established, refill the burette and slowly add the titrant drop by drop when it is nearing the endpoint. When you are in a 0.5 milliliter range of the endpoint, take the wash bottle and transfer fractions of drops from the burette tip into the solution. Repeat the procedure 3 times or more until 3 precise titers are obtained. If one titer is significantly different from the others, ignore it and replace it with another titration. Calculate the average of each titer and proceed to calculating the concentration of the solution of the unknown concentration.